Hello and welcome dear students. Today we are going to discuss the topic RNA processing. The main objectives of today's lecture are to know the basic about RNA molecule, to study the processing of RNA, to know about the types of RNA processing, to know about significance of RNA processing. Dear students, let's first start with the introduction to RNA. Ribonucleic acid or RNA is a linear molecule composed of 4 types of smaller molecules called ribonucleotide bases, that is guanine, uracil, adenine and cytosine. That means in RNA, instead of thymine which is present in DNA, there is uracil. Unlike DNA, RNA is usually single-stranded. Additionally, RNA contains ribose sugar rather than the deoxyribose sugar, which makes RNA more unstable and more prone to degradation. Each ribonucleotide base consists of a ribose sugar, a phosphate group and a nitrogenous base. Adjacent ribose nucleotide bases are chemically attached to one another in a chain via chemical bonds called phosphodiester bonds. RNA is synthesized from DNA by an enzyme known as RNA polymerase during a process called transcription. The new RNA sequences are complementary to their DNA template rather than being identical copies of the template. So, RNA is often compared to a copy from a reference book or a template because it carries the same information as its DNA template but is not used for long-term storage. RNA is then translated into proteins by structures called ribosomes. In transcription, the DNA sequence of a gene is rewritten in RNA. In eukaryotes, the RNA must go through additional processing steps to become a messenger RNA or mRNA. In translation, the sequence of nucleotides in the mRNA is translated into a sequence of amino acids in a polypeptide or protein chain. Genetic code is the mechanism by which genetic information is stored in living organisms. The code uses sets of 3 nucleotide bases, that is codons, to make the amino acids that in turn constitute proteins. So, we can also define the genetic code as the set of correspondences between the codons of the mRNA and the amino acids of the proteins produced on the ribosomes through the translation of these mRNAs. The genetic code is of 2 types. The genetic code can be expressed as either RNA codons or DNA codons. So, RNA codons occur in messenger RNA, that's mRNA, and are the codons that are actually read during the synthesis of polypeptides. The process is called translation. Dear students, let's discuss the types of RNA in a cell. In both prokaryotes and eukaryotes, there are 3 main types of RNA. There are messenger RNA, that's mRNA, tRNA, that is transfer RNA, and ribosomal RNA, that is rRNA. These 3 main types of RNA are involved in protein synthesis. The RNAs of a ribosome are called rRNAs. tRNAs constitute a 3rd major class of RNAs that is required during protein synthesis. Both rRNA and tRNA owe their activity to their complex secondary and tertiary structures. Many RNAs fold into complex 3-dimensional shapes. The mRNA is known as coding RNA, while all other RNA types are known as non-coding RNAs. A non-coding RNA, or an ncRNA as it is abbreviated, is a functional RNA molecule that actually skips this last step and is not translated into a protein. In other words, they just go directly from transcription into an RNA molecule and then go off to perform any number of vital functions within the cell. There are many examples of non-coding RNAs, including microRNAs, ribosomal RNAs, transfer RNAs, the list goes on and on. Dear students, let's now discuss all of them one by one. First, the transfer RNA, abbreviated as tRNA. This is a small RNA molecule that plays a key role in protein synthesis. Transfer RNA serves as a link or adapter between the messenger RNA mRNA molecule and the growing chain of amino acids that makes up a protein. Now let us discuss the ribosomal RNA or rRNA. This RNA molecule in cells that forms part of the protein synthesizing organelle known as ribosome, and that is exported to the cytoplasm to help translate the information in messenger RNA, that is mRNA, into a protein. Dear students, let's discuss the smaller nuclear RNA, that is snRNA, located in the nucleus, involved in RNA splicing, maintaining telomeres, etc. The another one is smaller nuclear RNAs or snRNAs, small RNA molecules that guide chemical modifications of other RNA, rRNAs, tRNAs, and snRNAs. Now let us discuss the small interfering RNAs or siRNAs. This is a class of double-stranded RNAs, 20 to 25 nucleotides in length. siRNAs are involved in the RNAi pathway where they interfere with the expression of a specific gene. Another class is microRNAs or miRNAs. These are single-stranded RNA molecules of 21 to 23 nucleotides in length. Their main function is to downregulate gene expression. Our genome encodes hundreds of these tiny microRNAs. Dear students, let's now discuss the RNA processing. Transcription is the mechanism by which the information in genes, that is DNA, is used to produce RNA. The newly made RNA, also known as the primary transcript, the product of transcription, is known as transcript or pre-mRNA, is further processed before it is functional. So transcripts that encode proteins will proceed to the ribosome for translation. In bacterial cells, mRNA can be translated directly as it comes off the DNA template. In eukaryotic cells, however, RNA synthesis, which occurs in the nucleus, is separated from the protein synthesis machinery, which is in the cytoplasm of the cell. In addition, eukaryotic genes have introns, which are non-coding regions that interrupt the gene's coding sequence. The primary RNA copied from genes containing introns will also therefore have regions that interrupt the coding sequence of the gene. These regions must be removed before the mRNA is sent out of the nucleus to be used to direct protein synthesis. The process of removing the introns and rejoining the codon sections or exons of the mRNA is called splicing. Proteins that interact with the cap, that is mRNA, and poly A tail allow the mature mRNA to be exported through nuclear pores into the cytoplasm for translation or protein synthesis. In prokaryotes, all steps in gene expression take place in the same cellular compartment, and thus transcription and translation of an messenger RNA are intimately linked, while as in eukaryotes, the nuclear membrane provides a physical barrier between the compartments in which an mRNA is synthesized and translated. During its sojourn in the nucleus, the primary RNA transcript undergoes many physical transformations, including 5' capping, removal of internal non-coding sequences via splicing, and addition of a 3' poly A tail. RNA processing is the term collectively used to describe the sequence of events through which primary transcript from a gene acquires its mature form. Transcripts from nuclear protein coding genes acquire a 5' cap structure very soon after their synthesis begins by RNA polymerase II. At the 3' end of the messenger RNA, a long string of adenosines is added in a process tightly linked to transcription termination. Finally, maturation of most eukaryotic mRNA precursors requires a process known as splicing, in which introns, the internal non-coding segments, are removed and the coding segments known as exons are joined to produce functional mRNAs. In complex eukaryotes, exons are much smaller than introns and provide the functional unit initially recognized by the splicing machinery. Because these splicing signals found at the exon-intron boundaries have low information content, ancillary elements known as splicing silencers and enhancers are required to specify the precise site of exon joining. The word intron comes from the word intervening. These are the intervening sequences that break up the coding sequence. You can remember that introns are in the way of the coding sequences. The word exon comes from the word expressed. These sequences are present in the mature mRNA. Throughout their life cycles, mRNAs are decorated with proteins, some associated only transiently, while as others bound stably. Thus, the functional form recognized by both the RNA processing machinery and the translating ribosome is a ribonucleoprotein complex, that is mRNP. An important advance in recent years is the discovery that RNA processing events are mechanistically coupled to transcription by RNA polymerase II. The capping, that is the enzymatic addition of a 7-methylguanosine not encoded in the DNA to the 5' end of the transcript, occurs very early in the biosynthesis of mRNA. This modification confers resistance to nucleases that degrade RNA in a 5'3' direction and also facilitates translation, that is the protein synthesis. The poly A tail, which can reach lengths of several hundred nucleotides in mammals, is also not encoded in the DNA. Like the cap structure, the poly A tail enhances RNA stability and promotes translation in the cytoplasm. These functions require binding to the major poly A binding protein designated PAB1 in yeast and PABPC in mammals. The third major nuclear event, pre-mRNA splicing, is the process by which internal non-coding segments or introns are removed from the primary transcript through a concerted mechanism that concomitantly joins the segments that form the mature mRNA, that is the exons. Finally, some pre-mRNAs are subject to editing, a process through which the sequence encoded in the DNA is altered in the RNA. The ultimate fate of an eukaryotic mRNA is to be degraded, that is turned over, which generally occurs in the cytoplasm after it undergoes multiple rounds of translation or protein synthesis. Throughout its life cycle, a given mRNA is subject to quality control processes known as RNA surveillance, which detect aberrant processing or damage and target non-functional RNA molecules for elimination, in some cases before they can be exported from the nucleus itself. Dear students, let's now discuss in detail the steps of RNA processing. Transcription is a mechanism by which the information in genes, that is DNA, is transcribed into RNA. The newly made RNA, also known as the primary transcript, that is the product of transcription, is known as transcript, is further processed before it is functional. Both prokaryotes and eukaryotes process their ribosomal and transfer RNAs. The initial product of transcription of a protein coding gene is called the pre-mRNA or primary transcript. After it has been processed and is ready to be exported from the nucleus, it's called the mature mRNA or processed mRNA. In eukaryotic cells, pre-mRNA undergoes three main processing steps, that is capping at the 5' end, addition of a poly A tail at the 3' end, and splicing to remove the introns and joining of exons. Let's first discuss the capping. In the capping step of mRNA processing, a methylated guanosine 7-methyl-G is linked to the phosphates at the 5' end of the mRNA. The cap protects the 5' end of the mRNA from degradation by nucleases and also helps the position of mRNA correctly on the ribosomes during protein synthesis. The 5' cap has four main functions: regulation of nuclear export, prevention of degradation by exonucleases, promotion of translation, that's ribosome, and translation in the cytoplasm. Promotion of 5' proximal intron accession. Dear students, let's now discuss the addition of a poly A tail at the 3' end. The 3' end of an eukaryotic mRNA is first trimmed, then enzyme called polymerase A or polymerase adds a tail of about 200 A nucleotides to the 3' end. There is evidence that the poly A tail plays a role in efficient translation of the mRNA as well as the stability of the mRNA. The cap and the poly A tail on an mRNA indicate that the mRNA is complete, that's not defective. Dear students, let's now discuss another process, that is the splicing of RNA processing. In many cases, splicing now appears to occur as the transcript is being synthesized. Introns are removed from the pre-mRNA by the activity of a complex called the spliceosome. The spliceosome is made up of proteins and small nuclear RNAs, that's snRNAs, that associate to form protein RNA enzymes called small nuclear ribonucleoproteins or snRPs. The splicing machinery must be able to recognize sequences that are specific to splice junctions, that is the end of each exon and the start of the next exon, in order to correctly cut out the introns and join the exons together to make the mature spliced mRNA, which is functional. There are two main steps in splicing. In the first step, the pre-mRNA is cut at the 5' splice site, that is the junction of the 5' exon and the intron. The 5' end of the intron then is joined to the branch point within the intron. This generates the variant shaped molecule characteristic of the splicing process. In the second step, the 3' splice site is cut and the two exons are joined together and the intron is released. RNA splicing facilitates the formation of multiple functional mRNAs from a single transcript which codes for different proteins. It also helps in the regulation of gene expression and protein content of the cell. So splicing makes genes more modular, allowing new combinations of exons to be created during evolution. Now let's have a detailed overview of the eukaryotic splicing. Eukaryotic genes that encode polypeptides are composed of coding sequences called exons. Exon signifies that they are expressed, and intervening sequences called the introns. Intron denotes their intervening role. Transcribed RNA sequences corresponding to introns do not encode regions of the functional polypeptide and are removed from the pre-mRNA during processing. It is essential that all of the intron encoded RNA sequences are completely and precisely removed from a pre-mRNA before protein synthesis or translation begins, so that the exon encoded RNA sequences are properly joined together to code for a functional polypeptide or protein. If the process errs by even a single nucleotide, the sequence of the rejoined exons would be shifted and the resulting polypeptide would be non-functional. The process of removing intron encoded RNA sequences and reconnecting these encoded by exons is called RNA splicing. Intron encoded RNA sequences are removed from the pre-RNA while it is still in the nucleus. Although they are not translated, introns appear to have various functions including gene regulation and mRNA transport. On completion of these modifications, the mature transcript, the mRNA that encodes a polypeptide, is transported out of the nucleus destined for the cytoplasm for translation purposes. Introns can be spliced out differently, resulting in various exons being included or excluded from the final mRNA product. This process is known as alternative splicing. The advantage of alternative splicing is that different types of mRNA transcripts can be generated, all derived from the same DNA sequence. In recent years, it has been shown that some archaea also have the ability to splice their pre-mRNA. The splicing reaction is catalyzed by the spliceosome, a macromolecular complex formed by the 5 small nuclear ribonucleoproteins SNRPs termed U1, U2, U4, U5 and U6 and approximately 200 proteins. The assembly of the spliceosome on pre-mRNA includes the binding of U1 snRNP, U2 snRNP, the preformed U4, U6, U5 triplet snRP and the PRP19 complex. This assembly occurs through the recognition of several sequence elements on the pre-mRNA that define the exon-intron boundaries, which include the 5' and 3' splice sites, the associated 3' sequences for intron excision, the polypyrimidine tract, and the branch point sequencing. In mammals, the first catalytic step of the splicing reaction begins when the U1 snRNP binds the 5' splice site of the intron defined by the consensus sequence AGG, URAGU, and the splicing factors SF1 and U2AF cooperatively recognize the BPS, that pyrimidine and 3' splice site to assembled complex E or the commitment complex as is shown in the figure. Subsequently, U2 snRNP and additional proteins are recruited to the pre-mRNA BPS to form the pre-spliceosome or complex A. The binding of the U4, U6, U5 tri-snRNP forms the precatalytic spliceosome or complex B. After RNA and RNA protein rearrangements at the heart of the spliceosome, U1 and U4 are released to form the activated complex B or complex B'. This complex is responsible for executing the first catalytic step through which the phosphodiester bond at the 5' splice site of the intron is modified by the 2-hydroxyl of an adenosine of the BPS to form the free 5' exon and a branched intron. The reaction of the 2-hydroxyl from the branch point adenosine nucleotide is known as transesterification reaction. During this process, additional rearrangements occur to generate the catalytic spliceosome or complex C, as is shown in the figure, which is responsible for catalyzing the second transesterification reaction, leading to the intron excision and exon-exon ligation. The resulting intron structure is referred to as a lariat structure. After the second catalytic step, the U2, U5, and U6 snRNPs are released from the post-spliceosome complex and recycled for additional rounds of splicing. Dear students, let's now discuss the alternate splicing. Many pre-mRNAs have a large number of exons that can be spliced together in different combinations to generate different mature mRNAs. This is called alternative splicing and allows the production of many different proteins using relatively few genes, because a single RNA can, by combining different exons during splicing, create many different proteins coding messages. Because of alternative splicing, each gene in our DNA gives rise, on average, to 3 different proteins. Alternative splicing may also be used to change or downregulate protein production from a gene by retaining intron that includes a premature stop codon, which will end protein synthesis before the final production can be completed. This role for alternative splicing may be useful for organisms that need to respond to a changing environment, such as temperature or light for plants. Profiling of mRNA after cold treatment in tea plants revealed that 28.8% of alternative spliced genes retained an intron, and this offers an additional mechanism for regulating protein production and function. Alternative splicing options are determined by the expression of or exposure to in trans elements present within unique cellular locations and environments. Additional sequencing elements within the mRNA, known as exonic and intronic splicing silencers or enhancers, participate in the regulation of alternate splicing. Specific RNA binding proteins, including heterogeneous nuclear ribonuclear proteins or HNRNPs and serine arginine rich, that's SR proteins, recognize these sequences to positively or negatively regulate alternative splicing as shown in the figure. These regulators, together with an ever-increasing number of additional auxiliary factors, provide the basis for the specificity of this pre-mRNA processing event in different cellular locations within the body. Dear students, let's now discuss the processing of tRNAs and RNAs after transcription. In addition to mRNA processing, non-coding eukaryotic RNAs are processed or trimmed, chemically modified from large precursor RNAs to mature functional RNAs. These precursor RNAs, pre-RNAs or primary transcripts contain in their sequences the information necessary for their function in the cell. Both prokaryotes and eukaryotes process their ribosomal and transfer RNAs. Pre-rRNAs, that is ribosomal RNAs, cleaved and or trimmed, not spliced because the pieces become separate RNAs to make multiple shorter mature rRNAs. Pre-tRNAs, that is transfer RNA, are trimmed. Some bases within the transcript are chemically modified, and 3' bases that is not encoded by the tRNA gene are enzymatically added to the 3' end. Dear students, it was all about today's lecture on RNA processing. Hope you have understood it well. Take care. Goodbye.